Hello guys, welcome to our channel. Today I will discuss about activation of receptor tyrosine kinase or RTK and MAP kinase signaling pathway in detail. Watch my previous videos where I have discussed upon GPCR mediated cyclic AMP pathway and inositol phospholipid signaling pathway in detail. Links of the videos are provided in the description. So let's start. Like GPCRs, enzyme coupled receptors are transmembrane proteins with their ligand binding domain on the outer surface of the plasma membrane. Instead of having a cytosolic domain that associates with a trimeric G protein, the cytosolic domain of enzyme coupled receptor either has intrinsic enzymatic activity or associates directly with an enzyme. Whereas a GPCR has 7 transmembrane segments, each subunit of an enzyme coupled receptor usually has only one. GPCRs and enzyme coupled receptors often activate some of the same signaling pathways, and there is usually no obvious reason why a particular extracellular signal utilizes one class of receptors and not the other. Now, there are 6 principal classes of enzyme coupled receptors. These are mentioned in this table. Many extracellular signal proteins such as epidermal growth factor, platelet derived growth factor, fibroblast growth factors, etc. are ligands that act through these enzyme linked receptors. At first, we will understand the receptor tyrosine kinase or RTK in detail, and then we will learn about the MAP kinase signaling pathway. So, the human RTKs are encoded by about 60 genes. These receptors can be classified into more than 16 structural subfamilies. In all cases, the binding of the signal protein to the ligand binding domain on the outside of the cell membrane enables the intracellular tyrosine kinase domain of the receptor to phosphorylate selected tyrosine side chains. Ligand binding causes the receptor chains to dimerize, bringing the kinase domains of two receptor chains together so that the two chains can cross phosphorylate each other on multiple tyrosine residues. This process is referred to as trans-autophosphorylation. This results in the activation of the receptor. The phosphorylation of tyrosines within the kinase domain increases the kinase activity of the enzyme. Also, phosphorylation of tyrosines outside the kinase domain creates high affinity docking sites for the binding of specific intracellular signaling proteins. Several intracellular signaling proteins can bind to the phosphotyrosines on activated RTKs. These intracellular signaling proteins that bind to phosphotyrosines either on the activated RTKs or on proteins docked on them have varied structures and functions. However, these proteins usually share highly conserved phosphotyrosine binding domains. These can be either SH2 domains, where SH2 stands for SRC homology region, or less commonly the PTB domains, that is the phosphotyrosine binding domains. So, the question arises, what is SH2 domain? The SH2 or SRC homology 2 domain is a structurally conserved protein domain found in the SRC oncoprotein and in many other intracellular signal transducing proteins. SH2 domain allows protein containing this domain to dock to phosphorylated tyrosine residues on other proteins. These SH2 domains recognize phosphorylated tyrosines and bind to them. Many signaling proteins also contain other domains such as SH3 domains that bind to proline-rich motifs of the intracellular proteins. These signaling proteins, after binding to the activated receptor, propagate the signal downward. However, some signaling proteins on the other hand give negative feedback, for example the CBL protein. This protein docks on the activated receptor and catalyzes their ubiquitination called monoubiquitination. This results in endocytosis and degradation of the receptor in the lysosome. This is an example of receptor downregulation. Mutation in the CBL protein can lead to prolonged RTK signaling leading to cancer. This results due to the fact that mutation in the CBL protein prevents the ubiquitination of the receptor. Some signaling proteins are composed almost entirely of the SH2 and SH3 domains. They are called adapter proteins. They couple the activated RTK to other downstream signaling proteins such as RAS that do not have SH2 domains. This is the crystal structure of a mammalian adapter protein GRB2. Now coming to RAS. RAS is a monomeric GTPase that can activate various downstream signaling pathways. It acts as a signaling hub from where different downstream signalings can take place. RAS remains anchored to the cytoplasmic phase of the plasma membrane and relays the signal through different proteins. RAS functions as a molecular switch, just like G-alpha subunit of the G protein. RAS is active when GTP is bound and inactive when bound to the GDP. Here we can see that the RAS guanine nucleotide exchange factors or RAS GEF proteins stimulate the dissociation of GDP, leading to the uptake of GTP from the cytosol. This activates RAS, while the RAS GTPase activating proteins or RAS GAPs increase the rate of hydrolysis of bound GTP and thus inactivates the RAS. RAS gap does not work on hyperactive mutant RAS. As a result, the RAS remains active for long period, leading to cancer. As we have understood about RTK, adapter proteins, SH2 and SH3 domains, RAS GEFs, RAS proteins. Now let's understand the MAP kinase signaling pathway. Here we can see the RTK receptor. Binding of extracellular signaling molecules activates this receptor. In mammalian cells, the adapter protein is GRB2 or growth factor receptor bound protein 2. This GRB2 has both SH2 and SH3 domains. Through the SH2 domain, the GRB2 interacts with the phosphorylated RTK, while through the SH3 domain, the adapter protein binds to the RAS GEF protein, which is called son of sevenless or SOS. Now this SOS performs the GEF function and removes the GDP bound to the inactive RAS. As a result, GTP from the cytosol automatically binds due to high concentration of GTP in the cytosol to the RAS. This activates the RAS protein. The active RAS further stimulates the SOS, giving the positive feedback. Due to the presence of RAS gap, the RAS signal is short-lived. It has been discussed in the previous slide. To carry the signal forward, the activated RAS activates a MAP kinase signaling module. First, the activated RAS recruits the protein RAF to the cytosolic phase of the plasma membrane and then activates it by phosphorylating a serine threonine residue. This activated RAF is called MAP kinase. The activated RAF in turn phosphorylates and activates the next protein called MEK or MAP kinase. This activated MEK now phosphorylates and activates the protein ERK or MAP kinase. This is the final kinase of the MAP kinase module. Now the activated ERK or MAP kinase enters the nucleus and phosphorylates one or more components of a gene regulatory complex, which activates the transcription of a set of immediate early genes. These genes are called immediate early genes because they express within minutes from the activation of the receptor. Some MAP kinase relays the signal downstream by phosphorylating various other proteins in the cell. So in this way, the RAS MAP kinase signaling pathway conveys the signals received by the RTKs from the cell surface to the nucleus and alters the pattern of gene expression. So today I have discussed about what is enzyme coupled receptor, how it is different from GPCR, detailed discussion on RTK and on the MAP kinase signaling pathway. Watch my previous videos where I have discussed upon GPCR mediated cyclic AMP pathway and inositol phospholipid signaling pathway in detail. Links of the videos are provided in the description. If you have any queries or suggestions, please leave a comment. Don't forget to like and share my videos and subscribe to my channel. Thank you. Stay home, stay safe and study online.